Hi everyone, this is Dr. Kuo, and today we'll be talking about muscle physiology. First of all, we'll begin by reviewing some anatomy on the different muscle tissue types, and then we'll look at a review of muscle microanatomy before we move on to talking about muscle physiology. So recall that skeletal muscle and cardiac muscle are the two striated muscle types. Smooth muscle is not striated. So skeletal muscle and cardiac muscle, you can see striation under the microscope. Skeletal muscle are also characterized by multinucleated cell, and they have these long cylindrical cell bodies. They have multiple different nuclei around their periphery. Cardiac muscle, on the other hand, generally have just one or up to two nuclei per cell. And their adjacent cells are actually joined with these intercalated discs. Okay. And together they form a branching pattern. Smooth muscle cells tend to be spindle shaped, and so their nuclei reflect that as well, are also spindle shaped, meaning they're tapered on the two ends. Now, in terms of location, skeletal muscles tend to be found attached to bones through tendons, sometimes directly attached to bones. And then cardiac muscles are only found within the wall of the heart. Smooth muscles are oftentimes found within hollow organs, like blood vessels, especially the larger blood vessels, and then in many parts of our digestive tract, okay, and many other hollow organs. And then skeletal muscle is the only voluntary muscle. The other two, okay, as opposed to being innervated by somatic motor neurons like skeletal muscle, these two are actually innervated by the autonomic nervous system. Okay, and some of these differences are summarized in this table. So go over it, and some of these details are things that we'll talk about later in this lecture. Now, let's take a look at a review of muscle anatomy. Looking at the way that skeletal muscle is structured and organized, we see that a muscle fiber, which is synonymous with a muscle cell, can be bundled to form a muscle fascicle. Okay, and individual muscle cells or muscle fibers are surrounded by endomysium. And so when you take a bundle of those skeletal muscle cells, they form a muscle fascicle, and that muscle fascicle is wrapped around by perimysium. Okay. And then when you take different muscle fascicles and bundle them together, okay, together they form a single muscle, and that muscle is wrapped around by epimysium. Okay, and then that muscle as a whole is then joined to bone through tendon. Okay, and then notice that you can tell the level at which the muscle fiber or muscle cell appears because of the nuclei, multinucleated characteristic of muscle cell. And those nuclei tend to be lying on the periphery of the cell. Okay. So very important you understand that muscle fibers is synonymous with muscle cell. Okay, when we talk about skeletal muscles, a muscle cell is a muscle fiber. And then skeletal muscle cell membrane, so cell membrane of the skeletal muscle cell, we have a special term for it called sarcolemma, where skeletal muscle cells cytoplasm is also called sarcoplasm. And then for the modified endoplasmic reticulum that's used for calcium storage, discussed later in this lecture, when we see these in skeletal muscle cells, we call them sarcoplasmic reticulum. Okay, so basically it's just a modified form of endoplasmic reticulum. Okay, and then make sure you understand how these terms are defined. So muscle fibers, as we mentioned, is the same as muscle cells. Okay, so this is a cross section of a segment of a single muscle cell. Okay, within individual skeletal muscle cells or with individual muscle fibers, we find these contractile organelles or contractile elements called myofibrils. And so this is a myofibril, okay, and these are individual myofibrils. These myofibrils are rod-like contractile elements that take up most of the cell's volume. And they actually are composed of sarcomeres that are lined up end to end, okay? And these sarcomeres are composed of different myofilaments. So myofilaments are basically these individual filaments that make up the myofibril. So myofilaments specifically are referring to the actin filament or the thin filament and the myosin filament, also called the, which are the myosin filaments. So thin filaments are actin filaments, thick filaments are the myosin filaments. Okay. So this alternating pattern of thick filaments, thin filaments, thick filaments, thin filaments creates a banded pattern that we see under light microscopy. Okay. And so once again, thick filaments are made of myosin, thin filaments made of actin. We also have accessory proteins like troponin and tropomyosin. Troponin and tropomyosin, and whose functions we'll discuss later in this lecture. And then titin and nebulin are proteins that hold the thick and thin filaments in their position. So nebulin, we've got titin. Okay. Now, you may see terms like G-actin, F-actin, and what they are, the G-actin are just globular actin, meaning they're spherical monomers that actually chain together or polymerize to form the filamentous actin or F-actin. Okay, so when you hear the term actin filament, it's talking about the F-actin, basically a polymer of the G-actin. Hope that makes sense. Okay. Okay, another special organelle in understanding muscle physiology is the transverse tubules. So these transverse tubules, shown here in yellow, are seen as invaginations or extensions of the sarcolemma or the cell membrane of the skeletal muscle cell. So they penetrate into the center of the skeletal muscle and also cardiac muscle cells. And these are areas of the cell membrane that invaginate and actually contain large density of ion channels, transporters, pumps, etc. And that allow for the rapid transmission of action potential to take place. And so for that reason, they are important for how we bring about muscle contraction in skeletal muscle cells. Okay, and these also actually happen to have an important role in the regulation of calcium concentration within skeletal muscle cells, something we'll discuss again later in this lecture. Okay, and so on the next slide, we'll now zoom into a segment of a myofibril to examine its various repeating regions. So this is the illustration illustrating different components of a sarcomere. So a sarcomere is defined as the region from one Z disc to the next Z disc. Okay, and within it, we can see at the center of the sarcomere. Okay, this is the M line. Okay, and we've got the thick filament or the myosin shown in purple, and then in red is the actin filament or thin filament. okay? And then we have different regions. So the A band is marked by the boundary of this myosin filament, okay? And then M line is at the center of the A band. H zone is marked by the end of this actin filament to the end of the next actin filament, okay? And then we also have the I band, which is marked by the end of this thick filament to the end of this next thick filament. Okay, so understand and these concepts and try to draw this out so that you understand what each of these regions really refer to. Okay, for example, that A band is really composed of a combination of myosin as well as actin, both thick filament and thin filament. H zone only has thick filament. I band only has thin filament, okay, etc. And so when our muscles contract, we see a shortening within sarcomere, okay? And you'll notice in the animations that we are about to watch that there is a shortening in I band and H zone, but the A band, its length stays the same. So let's take a look at these animations. So I've included a number of these animations we can take a look at. Here's one of them. So you can pause the video and drag down here to see this in slow motion. Okay, so during muscle contraction, you can see this is a single sarcomere from Z disc to Z disc, okay? And in the middle of the sarcomere, we see the M line, okay? And this is our thick filament. So this is our A band, okay? And then from one end of this thin filament to the end of this next thin filament, we have the H zone. And notice that as our muscle contract, we see a shortening in the H zone. Okay. Notice that the A band does not change in its length, right? A band is defined by the ends of the same thick filament, right? So, okay. Another thing that shortens is our I band over here, from here to here. Okay. And then just another animation that illustrates the same thing. Here, they are not illustrating the M line. Okay. And then we also have this one. Okay, so you can watch these in slow motion on your own. We're now ready to look at muscle physiology. Okay, and so muscle contraction can be broken down into a few separate phases. The first of which is events happening at the neuromuscular junction. And this is something we've already discussed in a previous lecture. Okay, and so recall that this motor neuron has an axon potential that travels down to axon all the way to its axon terminal. And at that axon terminal, the electrical signal gets converted to a chemical one, where there's calcium influx leading to docking of these synaptic vesicles and release of the neurotransmitter acetylcholine. Acetylcholine then crosses that synaptic cleft, and now that chemical signal is getting converted back into electrical one. Okay, so acetylcholine binds to acetylcholine receptors, specifically the nicotinic type of cholinergic receptors, and that causes these ionotropic receptors to open, and then there is a sodium influx potassium efflux, but since there's a net sodium influx, it causes depolarization in this postsynaptic membrane within this skeletal muscle cell. And so that depolarization in the skeletal muscle cell, okay, then spreads. And so this is where we enter the second phase, the excitation contraction coupling. Okay, and so in summary, the EC coupling, excitation contraction coupling, referring to the events where that depolarization and spread throughout the sarcolemma of the skeletal muscle, eventually leading to calcium release from intracellular stores, specifically the sarcoplasmic reticulum. And then that calcium release then allow for its binding to troponin, which then through detailed events, which we'll discuss later, allow for actin and myosin to interact in such a way to lead to muscle contraction. So that's our last phase, the contraction relaxation cycling. Okay, that interaction between thick filaments and thin filaments. Okay, and then this last phase, okay, termed contraction relaxation cycling, is based on the sliding filament theory. Something again we'll discuss in more detail later, but basically is the theory of contraction that states that during contraction, we see overlapping thick and thin filaments that slide past each other in an energy dependent manner as a result of the actin myosin cross-bridge movement. Okay, again, these are details that we'll be exploring later. And so this diagram over here shows a summary of those separate phases. So we'll first be reviewing the events happening at the neuromuscular junction. Keep in mind, this is the topic that we've already discussed in a previous lecture, so it's nothing new. So here I've included an animation or a video. You should pause our lecture video and watch this video now by clicking this link. We now move on to talking about the excitation contraction coupling phase, or the EC coupling phase, as well as the calcium signaling that follows. Okay, so as we've just discussed, we got motor neuron releasing acetylcholine into synaptic cleft, and the acetylcholine binding to postsynaptic membrane on the sarcolemma. And then the net sodium influx causes depolarization, and that actually initiates the action potential within skeletal muscle membrane. And then we got an action potential that then travels, it's spread throughout the sarcolemma, including into these invaginations called transverse tubule or T-tubule. And then as the action potential spreads down the T-tubule, it triggers these voltage-gated calcium receptor channels called the DHP or dihydropyridine receptors located on the T-tubules. Okay, these DHP or dihydropyridine receptors, also called L-type voltage-gated calcium channels, are actually linked to these other sets of channels located within or on the membrane of sarcoplasmic reticulum. And these other set of calcium channels are known as ryanodine receptors. So basically we've got the DHP receptor and our ryanodine receptors that are linked to one another. And action potential spreading down T-tubule, that voltage from the action potential triggers a conformation change in the DHP receptor, which then opens up the ryanodine receptors. So then what follows is calcium release from sarcoplasmic reticulum into the cytosol, into the cytoplasm. Calcium then is able to bind to troponin, and troponin, being complex to tropomyosin, then tugs on the tropomyosin and moves it and shifting it. And then we'll look at the detailed events later, but what happens then is the movement and or the shifting of the tropomyosin then allows for interaction between the actin and myosin filaments. Okay, and then the myosin filament then can carry out the power stroke. Again, something we'll look in more detail later. So this is during the contraction phase, during the excitation contraction coupling. During the relaxation phase, on the other hand, the way that muscle relaxation is able to be initiated is when calcium in the cytosol is now pumped back into sarcoplasmic reticulum through the calcium ATPase. Okay, so following that, there's a drop in the cytosolic concentration of calcium. Okay, and with a drop in calcium concentration in cytosol, calcium will unbind from troponin, and then the tropomyosin will then shift once again, but now covering up the myosin binding sites on actin. Okay. So I've included a link to a great animation that you can now pause our lecture video to watch. This is a closer look at what we just discussed in the previous slide. So during the excitation contraction coupling, calcium released from sarcoplasmic reticulum binds to troponin. Troponin, being complex with tropomyosin, then is able to cause the tropomyosin to shift and thereby exposing the myosin binding site on the actin. So now actin and myosin are now able to form a strong connection and interact. Okay, and then the power stroke happens. Okay, and power stroke is something we'll talk more about in the next phase, the contraction relaxation phase. Okay, that's the last phase of the muscle contraction. Okay, so anyway, during the relaxation phase, on the other hand, we get calcium being pumped back into the sarcoplasmic reticulum. And now with the drop in cytosolic calcium, calcium unbinds from troponin, and tropomyosin again covers up that myosin binding site on actin. Okay, and so during this relaxed state, the interaction between myosin and actin is weak. Here we have a video on excitation contraction coupling. It's a summary of what we just discussed. So go ahead and watch this video by clicking this link. This graph here shows the timing of electrical and mechanical events that happens during our excitation contraction coupling. And so what we see here is the action potential of a somatic motor neuron. And that's followed by the skeletal muscle action potential. So you can see the two happens with a little delay. However, when we compare the timing of the muscle fiber action potential to the timing of the development of tension that happens during a muscle twitch, then we see that there is a latent period, which is that delay between when action potential begins and versus when the tension begins to develop in the muscle. Okay, and this delay actually represents the time that's required for calcium release from sarcoplasmic reticulum, as well as binding to troponin. The calcium is binding to troponin. And here in this graph, you can see that this graph is showing development of tension during one muscle twitch. A muscle twitch is defined as a single contraction relaxation cycle. So we've got a contraction and relaxation phase. So this is one muscle twitch. Okay. We now move on to talking about the last phase of muscle contraction, and this is the contraction relaxation cycle, and it's based on the sliding filament theory. And so here I've included a video or an animation that you can watch by following this link. Now, in this sliding filament theory, this is a theory that explains how our muscle can actually contract and generate force without creating movement. And so an example would be you pushing against a wall. And so there's clearly tension that develops within your muscles, because you can feel it, but you are not moving the wall. Okay. And so according to the sliding filament theory, the tension that's generated in our muscle fiber is actually directly proportional to the number of cross bridges that's formed between the actin and myosin filaments. And so here, we're going to take a closer look at the steps involved in contraction relaxation cycle. We begin with the rigor state. Okay. And so the rigor state is where the myosin heads are tightly bound to the actin filament. Okay, and in this state, there is no ATP or ADP bound to myosin. And in living muscle, this rigor state occurs only for a very transient period, okay, before the next phase in the cycle, where ATP binding to myosin head causes myosin to be released from actin. So myosin head detaches from actin. Okay. And then what follows is, because myosin is an ATPase, a myosin ATPase, it's able to hydrolyze ATP into ADP and phosphate, inorganic phosphate. Okay. And so the energy coming from ATP hydrolysis allows for the myosin head to rotate and actually reattach to the actin filament, but now in a different location. Okay, it used to be attached over here, but now the new attachment point is about 1 to 3 actin monomers away from the original position. So what we see here is the energy released from ATP hydrolysis is now trapped and stored as potential energy in the form of the rotated myosin head. Okay, so this rotated myosin head is now set to be in a cocked position, or it's ready to rotate. And notice again that in a cocked position, the myosin is now binding to actin that's about 1 to 3 molecules away from where it started. Okay, and this potential energy stored in the angle of myosin head with its filament, as we'll see, is later going to be turned into kinetic energy through this next step of power stroke. So in this next step, the power stroke, with the calcium signal, which we discussed earlier, calcium being released from sarcoplasmic reticulum, and then binding to troponin, shifting troponin out of the way, or shifting tropomyosin out of the way, and revealing the myosin binding sites on actin. Okay, and now with actin and myosin now able to interact. Okay. In this step, you see that ADP and phosphate continues to remain bound to myosin. And then what happens next is phosphate is then released from the myosin head. Okay, and the release of phosphate actually allows the myosin head to swivel. And so this is the power stroke. Okay, so the head swings toward the M line, and then thereby sliding the actin filament along with them. Okay. And then following this, the myosin then releases ADP. So first it releases phosphate, then it releases ADP as well. Okay, so myosin release from ADP causes the myosin head to again tightly bind to the actin filament and to return to the rigor state. So rigor mortis is basically a prolonged period of the rigor state, and this happens when we die. Rigor mortis is the stiffening of the limbs after we die, and the cause for it is our cells unable to carry out cellular respiration now is no longer generating ATP. Okay, and so with the level of ATP dropping in the cells, the myosin is no longer able to detach itself from actin. Okay, because without ATP binding, myosin will just remain bound to actin. And so what results is that antagonistic muscles, for example, biceps brachii and triceps brachii, are no longer able to change length, and so the body remains in this stiff position. Here's a link to yet another video. In this lecture, you'll notice that it really helps to watch these animations to help you envision what's really going on in the process. So go ahead and stop our lecture recording and watch this video as well. Let's now talk about the length tension relationship. So this length tension relationship describes that sarcomeres is able to contract with optimal or maximal force when it's at the optimal length. That means neither too long nor too short. Okay, and so if you look at this diagram, you'll see that there is a point at which there is optimal or the maximal amount of overlap between the actin and myosin filaments. Okay, when these two increase in their length, or I mean, when the sarcomere increases in length, you'll notice that gradually there begins to have a drop in their amount of overlap. And something happens when you go down on this end of the curve. So there is an optimal point which these two have the maximal amount of overlap. And at this point, this is when we're able to generate the maximum amount of force. Okay, and when we apply this length tension relationship to myocardium, so to the heart muscle cells, this is when we call this Frank-Starling relationship. Okay, and this is a term we'll hear in a later chapter. Let's now take a look at summation of muscle contractions. In the first part of this diagram, we see single twitches representing stimuli that are separated quite a bit in time. And so the muscle is allowed to relax completely between the different action potentials. Okay. And then in the second part of the graph, we've got two stimuli that are closer together. And so the muscle doesn't relax fully after the first stimulus before the second stimulus arrives. And so there's a summation effect. Okay. And so the buildup of tension from the two stimuli is going to be greater than what's generated from a single stimulus. And then in this third part of the graph, we see what's called an infused tetanus. Okay, and so in this infused tetanus, there is a series of repeated action potentials that are separated by a good amount of time between the stimuli. Okay, and so as a result, the muscle fiber has a bit time to relax partially before the next stimulus arrives. Okay, so tension is able to be accumulated through summation, but you can see there is this infused tetanus at the maximal tension state. And this last part of the graph, okay, shows when action potential continues to stimulate the muscle fiber repeatedly at a high frequency. And so relaxation between the contractions is not allowed to occur. And so here the muscle fiber is able to achieve a state of maximal contraction called the tetanus. Okay, and at the end of this tetanus, we see a decline in muscle tension, and this is due to muscle fatigue. So muscle fatigue happens when the muscle loses tension despite the continuation in the stimulus. Okay, so infused tetanus versus complete tetanus. And by the way, infused tetanus is basically a term for incomplete tetanus. Okay, infused tetanus is the same thing as complete tetanus. Moving on to talking about motor units. So a motor unit consists of one motor neuron together with all the muscle fibers that it innervates. Here we see a muscle, and we understand that skeletal muscle cells are long cylindrical cells, also known as skeletal muscle fibers or muscle fibers. And these muscle fibers can be grouped into muscle fascicles. And then if we group the muscle fascicles, we get the whole muscle. Okay. And we can see here that even within a single muscle fascicle, the muscle fibers can be innervated by different neurons. So each muscle is served by at least one motor nerve, not to be confused with axon, right? Because each motor nerve contains axons of up to hundreds of motor neurons. Okay, so that means we can have hundreds of neuronal axons coming to innervate this muscle. Okay, and as an axon enters a muscle, it branches into a number of endings, each of which then forms a neuromuscular junction with a single muscle fiber. So a motor unit consists of that one motor neuron, as well as all the muscle fiber that it controls or it innervates. Here we see a total of 2 motor neurons being illustrated. And so when that motor neuron fires and transmits an action potential, it will cause all the muscle fibers that it innervates to contract. Okay, and the number of muscle fibers per motor unit may be as high as several hundreds or as few as 4. Actually, in some cases, it can be up to thousands. And muscles that exert finer control, such as those controlling your eyes, your eye movement, as well as your fingers. Your fingers are able to carry out very fine movements. These muscles tend to have very small motor units. Okay. And by contrast, larger the weight-bearing muscles of your lower extremities, whose movements are less precise, these tend to have large motor units. The muscle fibers within a single motor unit generally are not clustered together, but instead are spread throughout the muscle, as we can see here. Right? These nerve endings or neuron endings, branches, are not innervating fibers that are right next to one another. Okay. So as a result, stimulation of that single motor unit causes a weak but uniform contraction of the muscle. Okay. This slide talks about the same thing, what we just talked about. And so the motor unit concept also allows for our motor units to act as a relay team. That means they can take turns serving the function. Okay, so some motor units can rest while others work to prevent fatigue.